Greetings everyone. My name is Shoipal Rai and I am the current biology faculty from Nucleon Coaching Institute. And with this topic itself, or with this video itself, we will be continuing with the series that I have created, that is Principles of Inheritance and Variation. And in today's topic we will be learning about what is linkage, because as I have told you, Mendel was the father of genetics, and as such he concluded two major laws. Two major laws that he gave, that were law of segregation and law of independent assortment. Although law of segregation is called as the law of universality, but law of independent assortment, however, has its exception, that being linkage. So we will begin our lecture with what has been. As you can see in this, in this we will be simply talking about how linkage works, what is linkage, and before knowing that, a bit of the basics on what you need to understand is that what happens during your linkage, or what happens during the usual gamete formation. Why? Because Mendel's law, that is law of independent assortment, law of independent assortment works best, or works it works when we discuss about gamete formation. Remember, both of these laws, that is law of independent assortment and law of segregation, both work when we discuss about gamete formation, or when gamete formation is taken into consideration. In that sense, what happens actually during gamete formation? During gamete formation, there may be a chance event that is called as crossing over. Something that is responsible for the mixing of the characters of both your parents. How does this happen? Let's assume that this blue colored chromosome and this red colored chromosome are part of chromosomes from two different parents. That is, the blue color could be represented by the male counterpart, whilst the red color one could be for the female counterpart. Now you can see I have written chromosome 1 and chromosome 1 in this. Why is such so? Because in this gamete formation, what is happening actually is that the diploid content of those cells is now being converted into your haploid contents. Now what did I mean by that is, that diploid, diploid is the condition represented by 2n. Diploid is the condition wherein we possess a complete extra set of unique chromosomes. What do we possess? We possess a complete extra set of unique chromosome. That means if you had chromosome 1, you will have another pair of chromosome 1. Both contributed through either your father or your mother. Both of them contribute to form something called as, what do a union of such a thing create? It creates zygote. It creates zygote, which is 2n. This alone, okay, when we talk simply alone, then we call it a haploid, okay, then we call it a haploid. Is it okay? Is it easy enough to understand what is diploid and what is haploid and how does this process begin? Okay. So with this small conclusion or with this small understanding, let us move forward. Okay. What happens actually in your crossing over? What actually does happen is that this okay, this is what happens during a crossover. Okay, that is the segments of the chromosomes that are called as chromatids. These are called as chromatids. Okay, I'm simply giving you all the information you need before we proceed further with linkage. Okay, these chromatids. Okay, and can you tell me what kind of chromosomes these are? These are identical chromosomes. Okay, that means the same pair of chromosome. Hence, it is called as homologous chromosomes. Okay, these are called as homologous chromosomes. Okay, what is actually exchanged? The exchange is the segment. Okay, the segment of these chromatids. Okay, just a part of these segments are exchanged. Okay, and when this exchange takes place, we call this to be crossing over, or recombinance or recombination. Okay, the product that they give is recombinants, but in this case we just simply talk about crossing over or recombination. This process is called as crossing over or recombination. Okay, now why did we study about this? What purpose does it serve for you to understand about linkage? Well, linkage, the term itself, okay, linkage, the term itself simply means the association. Why? Because linkage is itself association. Okay, association of genes, in turn alleles. Okay, what is linkage? Linkage is simply the association of genes slash alleles. Okay, in this, when we are exchanging, okay, when we are exchanging, what does actually your law of independent assortment say? What does the law of independent assortment? I'm simply writing IA. Okay, understand that this is independent assortment that we are referring to. Okay, what does this simply say? That during gamete formation, the frequency of gametes formed must not be influenced by the allelic pair slash genes. Okay, that simply means that when they are going through gamete formation, they must not interact in a way that simply changes the frequency of the formation of gametes. Okay, now how best to make you understand? Okay, we can simply take, okay, we can simply take an example. Okay, In your book, it is given about the bar-eye effect, or the basically linkage effects in case of Drosophila. Okay. Why? Because the scientist that worked on linkage the most was T.H. Morgan. Okay. Who is this T.H. Morgan? He is called as the father of modern genetics. Modern genetics and linkage. Okay. Why? Because he was the one who gave a proper understanding of linkage through work, or through his work on Drosophila melanogaster, that is fruit fly. Okay. By working on Drosophila melanogaster, he gave the idea of linkage by seeing the eye color, okay, and the wing shape. Okay, this is the example, I suppose, that is given in your book also. Okay, that is eye color and wing shape. Okay, what happens in this is, in the eye color part, okay, eye color part, you would find, usually you would find that there is your wild type and your mutant type. Okay, wild type and mutant type. Now what is this wild and what is this mutant? Wild simply refers to dominant and mutant simply refers to recessive. Okay, no need to get confused what is wild and what is mutant. It is simply dominant and this is recessive. So what is the trait for eye color? In this case, the dominant trait would be red. Okay, and the mutant trait would be white. Okay, red and white. Similarly, we would find for wing shape or body color also we can take. Okay, but I will go with wing shape, that is, you could get something called as notch wing. Okay, and you could get something as normal wing. Okay, so the normal one is your wild type and the notch is your mutant type. Okay, so if we take a cross of these two, then it will simply become a dihybrid cross, right? Why? Because we are taking two characters together. Now usually, okay, now usually what will be the expressed ratio in case of independent assortment? That means when we are assuming no linkage. Okay, in case of independent assortment, when you move to F2, okay, when you move to F2, we would find the ratio will always be 9331. We have already discussed how we reached on to the case of 9331. Okay, in case of phenotype, right, this is the ratio for a phenotype, right? But what must happen, okay, or what must happen in case of linkage is, in the same F2, okay, this ratio will no longer be 9331. It will change. Now what will it change into? Well, it can be 9:7, can be 9:3:4. Okay, it can be 9:3. It can be so many. Okay, but whatever it will be, it will never be this again. Why? Because when we consider linkage, the frequency the frequency of gametes formed is changed. Gametes formed is changed. And in doing so, we never get the expected outcome from the case of independent assortment. That is why linkage is the sole exception to the law of independent assortment. How does this work? How does this work? Simply because let us assume that we take the red eye color through capital R, or we show it through capital R, and the notch wing or the normal wing through N. Okay? What happens is that these two are two different genes. If they are present, on the same chromosome, whether or not in the same chromatid, that also matter, okay, but if they are present on the same chromosome, okay, or the same chromatid, they have a higher chances of being crossed over onto the next, okay, they have a higher chance of being crossed over. That means R and N have a higher chance of being crossed over or being inherited together. This is called as linkage. Okay, so linkage, I should say, okay, linkage is dependent on the distance. Oh, sorry. It is directly proportional to the distance. Okay, if the distance increases, okay, if the distance increases, linkage decreases. If the distance decreases, then linkage increases. Now, what is this distance? Distance between two genes. We are talking about the distance between two genes. Between two genes. Okay? When we talk about distance between two genes, if that is decreased, then linkage must increase, okay, and vice versa, okay. Now there is one thing that you must know, or that you should know about, is what happens when we talk about the products that we receive in the F2, okay. What happens is that we should get 9331 when we follow the normal case of independent assortment, okay. When we follow the normal case of independent assortment, we get 9, 3, 3 and 1, correct? But in case of linkage, this ratio is not observed. Why is that so? Because there is a condition, okay, when we will discuss on, or we have discussed on this last one, you find it to be recessive, correct? Recessive, fine. And this recessive is the same recessive that was used in making this cross. That means it is a part of the parent, or it is the same as parent, okay. This is same as the parent. And in here also, okay, and in here also you would find the same with a dominant one, the same homozygous dominant one, correct? The same homozygous dominant one for both the traits, right? This also homozygous dominant recessive. Both of these form parents, okay. So these are called as parental types, okay. These are called as parental types. If you are getting confused, let me clear it out. Let us take the example of round and wrinkled, okay. Round and green, okay. Let us take up the example that Mendel used in his experiment itself. that we got round and green, right? We take two characters, that is round and green. They will have their both alleles, that is dominant allele and their recessive allele, right? So for an F2, I will simply skip the F1 part. Okay. For F2, you will require, okay, my bad. For F2, you will require, for F2, you will require, for F2, you will require this. This is your F1 crossed with another F1, correct? Crossed with another F1, which should give you the types of gametes and the combination of those gametes that you require, correct? So I can simply do it like, it will be capital RG, it will be capital R small g, it will be small r capital G, and it will be small r small g, correct? Similarly for this, similarly for this, similarly for this, right? So I will simply say it is the same, okay? And I can simply write capital R R, capital G G, okay, capital R R, okay, and in this case capital G small g, okay. Same for this one, this will be capital R, no, it will be smaller, right, because both of these are same, okay. So this will become small r, capital G, okay, and then again small r and small g, fine. Out of this you found this, and in this same cross, okay, in the first line of the crosses that we have shown through Punnett square, we found our parental types, okay. This is obtained, that means this F1 is obtained from the cross of these type of parents, that is a homozygous parents, okay. These are called as parental types, while these are called as recombinants, okay. Why? Because these are not the same, okay, these are not the same as what you have expected from this or from these two, okay. This is deviated, right? This is not the same as this, or this is not the same as these two, okay. This creates recombinants and this creates parentals. Why did I make you understand this? Because this is required to understand that linkage can work in two phases, that is through forming parental types and through forming recombinants, okay. Parental types and recombinant types, okay. So what happens is that we talk about F2 onwards. Why do we talk about F2 onwards? Because from F2 things start to segregate, okay, or we call such kind of population segregating population. Why? Because in F1 you will always find them to be homogeneous. We will find them always to be homogeneous. That means not a single one of those crosses or not a single one of those progenies are different from each other. They are completely identical with the same set of genes, okay. But what happens in F2 is that we start to see combinations, we start to see further segregation, okay. And from F2 onwards we call the populations to be segregating populations. Why? Because we see a large diversity, okay. We see a large number or types of population, okay, like homozygous ones, heterozygous ones, all those combinations that you can arise from that Punnett square of your dihybrid cross in F2, okay. All those 16 types, okay. Each and every one, some of them are similar to each other, but most of them are different, okay. In this case also, when we talk about F2, in case of linkage, we have already discussed that it must not be 9:3:1. Why? Because if we assume linkage, this is never the case, okay. This is never the case. But what happens in these two types, that is parental and recombinants, it is simply saying that what happens in linkage can sometimes give you a higher count or a higher number of parental types, okay, and sometimes it can give you a higher number of recombinant types, okay. This depends on whether they are present on the same chromosome or same chromatid, or whether they are present on different chromatids but the same chromosome, okay. This receives recombinants and this receives parental. If there is a higher number of parentals, okay, if there is a higher number of parentals, that simply mean that the alleles or the genes itself, they do not want to separate, okay, and linkage is nothing but this want, that they do not want to separate, they do not want to make new combinations. So you don't get recombinants, okay, you don't get such high number of recombinants, you simply get a higher number of parentals in case. If this can be shown, or I can show it to you also, we can show this through a simple formula, that is this simply shows that half of to the power n, which simply gives you for the homozygous part, okay, and the heterozygous part can simply be written as just a moment, okay. Okay. What does this formula mean? 2n minus 1 to the power m. Okay. N and m simply mean that what kind of segregating generation and how many genes are we considering. N represents the number of segregating genes that we are considering, while m is the segregating population that we are considering. How many rounds of segregation has it occurred? Although this is not necessary for you to know, this simply gives you an idea that if we get one, if we take one, that means the first segregating generation, and if we take two genes, okay, if we take two genes, you should get three, correct? You should get 3. 4 minus 1 and to the power 1 will always result in 3. What is this 3? This 3 simply represents that what are the categories, or what are the categories to the types of genotype that you can get through this linkage, okay. And if you see in the cross itself, if you see in the cross, okay, not an issue. If you simply make a cross of yourself, or if you make a cross of it also, you will find that the maximum number of genotypes that you can receive from this is 3. That is, recombinants can be of 2 types and parentals, okay. Now what gives or what makes the recombinants be more and what makes the parentals be more, that I explained through a simple diagram through this, okay. If they are present on the same chromatid, there is a higher chances of parentals being formed more, but if they are present on different chromatids, then you get recombinants to be more, okay. Okay. So, I hope that this lecture was not very complicated and you are, or you have been able to understand a bit of linkage. If you need further explanations on how linkage works or what is linkage, okay, feel free to drop a comment, okay, and we will get back to you. Thank you for listening to us, and we will continue this series to the next part, which we will talk about the genetic disorders and we will be finishing it, and then we will start with some other topic. So take care and thank you for listening.